Okay, welcome back to another screencast. So today in class you all worked on your photosynthesis or light-dependent reactions puzzle, and I'm going to be talking about a few different pieces of that puzzle that are, well, I guess they're directly addressed in the puzzle, and this will just help make a few things a little bit more clear for you. So the first part of the screencast is going to be talking about chemiosmosis, and then we're going to take a break from talking about chemiosmosis and talk about photosystems for a little bit, and then we're actually going to jump back and keep talking about chemiosmosis, and you're going to see the relationship between these two things, chemiosmosis and the photosystems. So the first thing, chemiosmosis, there are two words here that you both are familiar with, osmosis being the movement of water, but in this case, chemiosmosis is the movement of chemicals. And specifically, we're going to be talking about the movement of H+ ions, or protons, across a membrane. But before we do that, I want to talk about a little analogy that is going to be important for you to understand and will help enhance your understanding of chemiosmosis. So let's dive right into that. So first, let's talk about a dam. So a dam, the purpose of a dam is to hold back water. So you take a stream or a river and you construct some sort of obstacle and build up a lake. But the dam isn't just a solid wall. There's actually going to be some sort of, some sort of tunnel through it. Oh man. So there's a little tunnel here, and inside the tunnel is going to be some sort of propeller that, as water moves through this propeller, it's going to spin. So this will spin around in a circle. And the propeller is going to be attached to some sort of generator, and as the propeller spins, it pushes electrons through a generator, and those electrons are then going to be transported down a wire to a local city or a town where they're going to be used for electricity. So as this spins again, electrons are going to be pushed through this wire. Okay, great. And the reason why water flows in this direction through the tunnel and out is because there's high potential energy here and low potential energy here. So there's a gradient driving the movement of this water. However, if we were to fill up this side also with water over here, there would be no more gradient and there would be no more drive. This is kind of like an isotonic solution. Things would still be moving through the propeller this way, but there would also be an equal and opposite movement through this way. So we would get no movement of electrons through our water. So in order to maintain our push, we have to maintain a gradient. So that concept, important for you to understand. Okay, we need to maintain that gradient. So let's talk about how that relates to cells. Okay, so first of all, let's discuss the chloroplast. Structure of a chloroplast. The chloroplast is actually composed of three membranes, three bilayers. So if this is our chloroplast here, there's an outer membrane and an inner membrane, and then there's another membrane system, which is where photosynthesis is actually taking place. So hold on a second and let me draw this inner membrane for you. Okay, so here's our fully drawn chloroplast. Notice here two membranes on the outside, both bilayers, and another bilayer on the interior. Now this is where photosynthesis is actually taking place. And these stacks have names. So each of the things, they kind of look like little jelly beans. This membrane is all these jelly beans are stacked together and they're contiguous with one another, meaning that there is this space in here, all in here. You can travel all the way through it without leaving the system. And one of these is called a thylakoid. So a stack of them would be a thylakoid stack, and it's also called a granum. So a stack of these things is a granum. And the thylakoids, this membrane, composes the thylakoid membrane system, or the TMS. Inside the thylakoid is called the thylakoid lumen. Remember we saw the ER lumen earlier. That just, lumen is a fancy word for the inside. So this inside space is the thylakoid lumen. And outside of the thylakoid, or the TMS, but still inside the chloroplast, this space here is called the stroma. Not to be confused with the stomata or stomates you saw on the bottom of a leaf. This is different, okay? The stroma is kind of like the cytoplasm of the chloroplast. Okay, so here's your structure of your chloroplast, and we're going to zoom in on a thylakoid stack, or a granum. I'm going to draw it below here. Okay, so here's our thylakoid, and the thylakoid is going to be composed of, or studying the thylakoid are going to be a bunch of proteins, some very important proteins. And, damn, I've drawn the thylakoid in green. I shouldn't have, because I want to draw, I'll draw it in a different color green. My photosystems are also in green. So here we have photosystem 2, PS2 and PS1, and then following the photosystem is going to be our ETS, or our electron transport system, and we'll talk more about what those are doing later. And then additionally, inside, or embedded within the membrane, is going to be a very important protein. It's a channel protein. So I'm going to draw it here as a type of pore. Okay, so this molecule, or this protein, is very important. It's called ATP synthase. ATP. And as you might have guessed, it's going to be responsible for synthesizing ATP. And we're going to talk in a second about how it does that. But, okay, going back to this. PS2 is the oxygen evolving complex on one side of it. So when light energy comes and hits this molecule, it actually catalyzes the photolysis of water. So if we have H2O in here, it's going to become H plus and O2, and we're also going to donate some electrons to the electron transport chain. So that's the main reason why we have this photolysis of water, is because we need electrons to excite and run down our electron transport chain. But also, very importantly, it releases this hydrogen right here and water. Sorry, hydrogen and O2. This O2 is just a waste product of the whole process. So it's actually going to just leave the cell and exit through those stomates, or those stomata at the bottom of the leaf. But this H plus is very important, and actually we have lots of photolysis of water, so we have a buildup of an H plus gradient within our thylakoid membrane system. So there ends up being a lot more protons, or hydrogen ions, within our thylakoid membrane system than outside. So very similar to our dam situation right here, where we had water at a high concentration on one side of our dam, which flowed through and generated electricity. We have a high concentration of hydrogen ions inside our granum, which are going to flow through ATP synthase, and in doing so, they're going to basically spin a little propeller on our enzyme, and they're going to take ADP, which is adenosine diphosphate, so 2 phosphates, plus inorganic phosphate found in the stroma, and catalyze the formation of ATP. Okay, adenosine triphosphate, which is going to be used by the cell to do useful cellular work. Okay, so this happens continuously, whoop, all this hydrogen that we've built up inside is going to leave, generating more and more ATP, but we're going to reach a point where the concentration of hydrogen outside the cell, or sorry, not the cell, outside the thylakoid membrane system is equal to the concentration of hydrogen inside the thylakoid membrane system. And then, just like if we filled water up outside our dam, we lose the push, we lose the impetus of movement through this enzyme. There's still going to be movement through, but it's going to go in both directions. Not helpful for generating ATP. So, hold on to that thought right now. We need a way to keep the concentration of hydrogen ions high inside our thylakoid, which brings us to our discussion of photosystems and how they work. Okay, so I'm going to segue for a second and then come back to this idea of keeping the concentration of hydrogen ions high inside the thylakoid. Okay, so photosystems are composed of molecules called chlorophyll molecules, and they kind of look like lollipops, like so, and they look like this, and they've got an organic part, which you all know by now means they're made of carbon. So they've got an organic part, and they also have an inorganic part. In the center of every chlorophyll molecule, there's a metal ion. Or a metal atom. And metals, as you remember from chemistry, I'm sure, are really good at both accepting and giving up electrons. They are easily oxidized and easily reduced. And in this case, this molecule, this chlorophyll molecule, is really good at being oxidized. Loses electrons, oxidation. Leo goes grr, loses electrons, oxidation. So these are very good at being oxidized. These metal ions are very good at giving up electrons. Another thing you should remember from your days in chemistry about electrons is if you have electrons circle a nucleus in orbitals. So if we have some sort of nucleus right here, and we have an electron that's going to orbit, and it's not really like a planet orbiting the sun. It's more complex. It's just it's a density. So there's a certain area which we expect to find an electron, but for the sake of this explanation, let's just keep it simple. So an electron orbits a nucleus in a certain area, its orbital, but if there's an input of energy, so say a photon strikes this electron, it can actually bump up an energy level into a new orbital. So this is now, a high, this is now a high energy electron, or a He, H-E-E, high energy electron. So, an electron can absorb a photon and go up, and conversely, it can also emit a photon and then drop back down. So, there's this relationship between absorbing photons, bumping up orbitals, and then emitting photons and dropping back down. Okay, so electrons have this property, and okay, so let's talk about, so a photosystem. It's composed of many different molecules of chlorophyll, like this one, and they're arranged in a particular fashion, like this. We have a bunch of them that form, sometimes you'll see it in a pyramid shape, sometimes you'll see it in a circle shape. I'm going to draw it in a pyramid because it's a little bit easier to understand what's going on in this orientation. Okay, so imagine we have our photosystem here. It could be photosystem 1 or photosystem 2. It doesn't really matter. But we have photons of light coming down and hitting these chlorophyll molecules at the top. Well, these chlorophyll molecules are going to absorb that photon of light, the electron in the metal ion in the center is going to bump up an energy level, and then they're going to drop down an energy level and emit a photon of light. So that happens in this row. Absorption, bump up an energy level, and then emission, bump down. But you'll notice this next chlorophyll molecule right here is going to be receiving photons from this one and this one. So there's like twice the amount of energy that this electron is receiving here because it's receiving a photon from here and here. So it bumps up to an even higher energy level. And so on and so forth. This is going to happen all the way down each row. So as we go down every row, we're concentrating the energy until we reach this last chlorophyll molecule, which is so important I'm going to put a box around it. It's either P700 or P680, depending on the chlorophyll, the photosystem that you're in. But this last chlorophyll molecule is going to receive all this energy, and instead of the electron just bumping up an energy level and then falling back down, it actually gets so much energy that it can kick an electron off and donate it to the electron transport system, or the ETS. So that's how a photosystem gathers light energy, focuses it in, and uses it to excite a high energy electron. Now, let's go back to our depiction of what's happening in our chloroplast again. Remember, the electron is donated, so every time this last chlorophyll molecule donates an electron to the ETS, it needs a new one. Where does it get it? Well, it gets it from water. So water is split, oxygen and hydrogen ions are produced, and we donate a low energy electron, which is then excited by our photosystems, by the light coming from the sun, and is kicked off into this electron transport system. Now, the useful cellular work that this electron does as it moves through this first photosystem is it actually pumps hydrogen ions back into the cell. This is not a cell, sorry, into the thylakoid membrane system. So as it passes down through here, it is going to pump hydrogen ions back inside the membrane, thus maintaining this gradient high inside the thylakoid and low outside, so we can keep producing ATP from our ATP synthase. So that's what happens in the electron transport system that follows photosystem 2. The electron transport system that follows photosystem 1 is a little different, but I'm going to leave you to figure that out yourself, given your puzzle. But both ATP and the molecule that is produced at the end of photosystem 1 are going to be used later on during the light-independent reactions, also known as the Calvin cycle. And we're going to talk about that sometime within the next few days. Okay, I think this is all you need to know for right now. Thanks for tuning in, and I hope this helps make the puzzle a little bit more clear.